Okay, so yeah, my name's Stephen Taylor. I actually work at the Wellcome Trust Centre of Human Genetics, and the sort of work I do is taking lots of data from lots of different modalities in biology and trying to integrate them together to make sense of them. And so this session here is really about how can AI help the scientific method, if you like. How is AI going to be better than us? Is it going to be collaborative? And how are we going to use it? Is it going to enhance our abilities? I think we're already seeing that it is enhancing our abilities, but we're hopeful that it won't sort of take over and continue to be a research help rather than sort of take over the world and murder us. So we're very lucky today. We've got some great speakers. We've got Shankar Srinivas. He's from Jesus College, and he's a professor of development biology. And his research is focusing on cell movements in embryos and the heart. And he's been doing lots of work in molecular time-lapse imaging, combining different modalities, proteomics, single-cell transcriptomics. And he's also does a lot of public engagement work. It was a meeting a couple of weeks ago about his work that he's collaborating with the digital humanities, looking at embodiment. But I don't think you're probably going to talk about that today, are you? So it's going to be pure science today, I think, right? More or less, okay, good. So I'll hand over to you, Shank.
Thank you for the introduction, Stephen. So I'm going to give you a little bit of an overview of some of the research work we do and our use of machine learning, but also computational, more broadly computational approaches to try to grapple with large sets of data and to visualize as well as to analyze and extract insights from these type of data sets. So in a broad sense, the main interest of my group is how the shape of the body emerges. The example I always give is that the chicken egg you might have had for breakfast looks nothing like a chick, but it will, if you just put it, incubate it at the right temperature, it will reliably give you a chick, and in the sense that it has a beak, wings, etc., which are not there in the fertilized egg. So these shapes emerge during development. And another aspect which I point out, it's obvious, but of course you never have a kitten hatching out of a chicken egg or a puppy. It's always a chick. So this is programmed into the material of the egg. And so it is amenable to investigation and to explanation. It's not magic. It is possible to be able to understand this process, however very, very complicated it might be. And that's what we work on. How you basically go from something like this. On the left is a fertilized mouse embryo. It's at the 2-cell stage. And at the right is a mouse embryo, somewhat later in development. In red, you can see the forming heart and the vasculature, the blood vessels. And you can see that towards the top will hopefully be recognizable as a brain. It's a naked brain at this stage. You don't have a head. And what are called what is the neck level? You have something called pharyngeal arches. They are what evolutionarily links us to fish, the gill slits of fish. So how do these shapes emerge? And that's what we work on in my group. Another example, just to drive the point home, this is a movie I made ages ago of a mouse embryo developing. And just to orientate you here, again, it's if you're not a biologist or even if you are an embryologist, it's hard to appreciate. But during development, we are a flat disc, roughly speaking, and various movements convert this flat disc into what is our body can be very crudely described as a tube from mouth to anus. And so what converts a flat disc into a tube? And it's these type of what are called folding movements. There are 3 types of movements: rostral, caudal, and lateral. And they fold a topologically flat disc into a tube. So if you think about it, you can go from the front, from the sides, and from the back, and you get a tube. And does anybody want to guess where they all meet? We still hold in our body the mark of where these different folds meet. Sorry? Sorry, what? Spine. Nope, they're in the front of the body. It's in the mark is in the front of the body. The belly button, exactly. They all come together, and that's what, and then they form the umbilical tube that connects us to the placenta. And we still bear the marks of it. Anybody who's not a cyborg or an android bears a mark of that. So this is a view of what's called rostral folding, the head side. And this is the other magical aspect of this, which really is what fascinates me and keeps me going in this business. The heart literally is in front of your brain during embryonic development. Okay? So it's this type of movement that brings the heart to its more what's called inferior position within the body, and you'll see that. So in this embryo at this stage, there are 3 tissues which are semi-transparent, so you can see all 3 of them in parts. The outermost tissue is labeled green transgenically, and you will see a shadow, this shadow that forms the heart. And the innermost tissue is a ridge, which you can hopefully see just behind the shadow, and that forms the brain. And when I play the movie, you'll see this type of movement, which brings, and you'll start to see the heart starting to, as it forms, starts to beat. It doesn't look anything like a heart, but that's the embryonic heart. You'll start to, this type of movement brings it down lower, and you'll hopefully you'll be able to see the rhythmic beating towards the end as it starts to spark into life. So we basically, again, try to study how this type of morphogenesis occurs. This movement, this shaping is the result of coordinated activity of the component cells. You can see individual cells in the outermost layer. They move in a coordinated manner to bring about this, these type of large-scale remodeling events, and we try to understand how that occurs. So for today, I'll tell you about one a specific question in my group, which tries to break this problem down. It's a simpler stage. At this stage, the embryo, the mouse embryo, looks like a cylinder. It looks nothing like a mouse. What we are studying here is the outer layer of cells, shown in green. That's to orientate you. And those cells start out at the tip of this cylinder, and then they move up one side of the cylinder, and those cells specify where your head forms. So that is they're an important group of cells. Their movement is important because if they don't move, your head will not form in the correct place, and the embryo terminates. It doesn't develop any further. This is a movie I made even longer ago about how this occurs, and you can see these cells moving. And here you're seeing a subset of cells. At this stage, the embryo has maybe 300 or 400 cells. Again, it's semi-transparent, so you can see interior tissues as well as exterior tissues. These are cells on the outside of the cylinder moving. And so we have always been fascinated by how these cells move, and we continue to be fascinated by them, and we still don't understand precisely how these cells move. To give you some context about the specific issue, these cells move within a field of cells. And so they need to negotiate their way through this crowded, packed, what's called an epithelial tissue. It's like if we were even more jam-packed in the room than we are now, and I had to make my way from here to that, the other end, I would need to shuffle my way across. And imagine we were interlinked arm to arm. I would need to release the arms with somebody else and then establish a linkage with another person and then make my way across the room. So how does that occur? There are no tears. When these cells move from one location to another, they don't leave a gap behind. They don't crawl on top of the cells ahead of them. The cells ahead of them get squeezed aside. So we wanted to try to understand the various component movements, the component behaviors that could contribute to this large-scale movement. And these are some examples of various limited attempts we've made at taking a quantitative approach to describe this behavior, looking at the extent of neighbor exchange, and you can see that there are some differences, looking at shape factor, which is a measure of how distorted a cell is. And again, you can see certain regional differences which might help explain, but it isn't a global view. So to take a global view, we took a collaborative approach, and these are the key players behind this. And I just wanted to point out Matthew Starr and Felix, who are both based here in Oxford. Felix has now gone to Texas, who are the prime movers behind this, with the contributions from others as well. What we did is use a type of imaging. This is dynamic movements occurring in a living embryo. So you need to be able to visualize it. And we used a type of microscopy called light sheet microscopy. I won't go into the details of it, but to say that it's very, very gentle. Just like you can get a sunburn and get hurt, embryos will die if you illuminate them to be able to visualize them. So you need to, people have developed lots of approaches of microscopy which are very, very gentle. So we used light sheet microscopy. To cut several years' worth of work short, we made three-dimensional volumes of this type of movement, which you can see at the top left of the screen, and that's the cylindrical embryo. And that involved developing computational approaches to actually fuse image volumes, because the light sheet microscope allows you to image embryos from different angles to get better resolution. It involved registering things, time registration, as well as spatial registration. And so essentially, we were able to record the movement of all the cells. Now, how do we deal with that in a quantitative approach? So we used mature maths to actually peel the surface of the embryo, and on the left you can see a rectangular projection, and at the right a polar projection of the surface of the embryo. And now you can see all the sides, aspects of the embryo. The anterior is the side that's facing you, or the head end. The posterior is the back end, and you can see all of them. You can see the individual cells, and hopefully you'll be able to see the movement of this subset of cells, which is directional. You can see the cells in front of them being distorted, and you can see a barrier to their movement. Look at these cells, they're rock solid. They don't move at all, and they prevent the cells that are the subject of this of our study, these cells, they prevent them from over-migrating. So now to then get a better grasp of what's going on, we turn to machine learning approaches. And again, these are by now very standard. These are things which we did 3 or 4 years ago. We used machine learning to actually enhance the quality, to augment the quality of the image data, because I explained how the light sheet microscope allows you to image embryos in a gentle manner. We're still struggling though with this balance of getting very high quality images, which you can use automated segmentation approaches on, versus keeping the embryo alive and healthy. So you're always working at the edge of the quality of your image. And so, for example, we created a convolutional neural network to be able to make increase the signal to noise of the data, lots of training, etc. And you can see it does a remarkably good job. And at that time, we played with 2 different individuals training it, creating 2 different training sets, human-based, obviously, training sets, trained the network on these 2 different training sets. And in fact, the 2 networks that were trained on different training data sets converged and gave you the same results. And they agreed with, the 2 networks agreed better with each other than the 2 humans did, which was at one level actually reassuring, that they were picking up the essence of what it means to be a cell membrane at that point. So we had this. Once we have the data like this, you can easily segment the cells, and once the cells are segmented, you can track them relatively easily. As I said, we flattened it for the purpose of visualization and for the purpose of being able to do the augmentation tracking, and also for of course, the manual curation of the tracked and segmented cells. But we retained 3-dimensional information. So now, having done all of this, we could wrap this back onto a mannequin, so to speak, of the embryo, recoup our 3-dimensional information, and get a virtual embryo. We've distilled the essence of the embryo. So now then to the quantitative approaches. We basically did this with 5 embryos with roughly 2,000 cells, and it's time-lapse, it's longitudinal data, so we had around 293 time points, which in total gives you around 90,000 data points. And so we needed to use some sort of approach to be able to handle this level of data. So we extracted various physical, morphological, and behavioral parameters, and the 2 molecular parameters pertaining to actin, which is one of the molecules that helps in their movement. And we used approaches which were unbiased hierarchical clustering to try to find patterns within these cells, to see whether there are certain cells in certain regions that show stereotypic behaviors or morphologies, etc., and therefore can help us explain how these cells move. This is, here we borrowed a trick from single-cell transcriptomic work, for people who are familiar with that, where there have been a lot of developments. It's really exploded on a beautiful computational approaches to be able to deal with large data sets of transcriptomic, epigenomic, accessibility type information, which pertains more to gene function. But we used those to cell behavior, those approaches to understand cell behavior. And this allows you to get what are called this UMAP representations here, where each dot is a single cell. Sorry, I could have just used my cursor here. So each of these dots are one of those 20,000, 90,000 cells, and which have been clustered according to behavior. And this is dimensionality reduction, so if it's multidimensional space, and it's been shown here in two-dimensional space. One of the key things about this is, of course, this is all non-destructive, so we know where each and every one of these cells came from. And we're able to plot them back onto the embryo, and we can actually see that this type of clustering in our what we call phenome, their characteristics, this abstract space, does correspond to anatomical space in the real embryo. And we can see in what way the cells change in their characteristics, the cells in different locations on this UMAP, as well as in the embryo. And just to to end, I will just briefly tell you about one key insight that we received from this. This type of approach told us that the cells that migrate have a high level of actin, which is this molecule which is involved in cell movements, amongst other things. And we were able to visually validate this looking at actual embryos. We were also able to see that most of the cell rearrangements took place in front of the cells that are important in specifying where the head forms. Okay? So the cells that are moving aren't doing it. And the analogy I gave in the beginning about interlinked people and having to let go of certain people and make new links, that isn't how the cells that move work. They just bulldoze their way through the crowd, and everybody else gets out of the way. And it's the cell, and that's in red and cyan. You see them undergoing the neighbor exchange. They're undergoing neighbor exchange so that they can get out of the way. They're also getting distorted. Their shape is changing. This is also validated by actual 2 more minutes. Perfect. That's my second last slide. So this is also validated again by looking directly at the mechanical properties of the cells, and it's a bit of a busy slide. I'll just summarize by saying we used a dye that allows you to visualize the mechanical tension of the membrane, so in living samples, and we could thereby tell that the mechanical tension of these moving cells is greater. The heat map, the more red you are, the higher your tension. We could tell that the tension is higher amongst the moving cells, and that this is actually genetically controlled. Coming back to this, what I spoke about in the beginning, about everything is in a sense is amenable to experimental manipulation and investigation, and is ultimately genetically controlled or by proteins. If you look at certain mutants which have a defect in the movement of these cells, the mutant in question is called lefty1, where the cells that specify where your head forms, they keep on migrating. They don't stop migrating. They migrate too much. You can see that in these mutants, there are aberrations in the distribution of mechanical tension, pointing to this type of tension being important in allowing the cells to migrate and also setting limits to the migration of the cells. So to summarize, the DV cells, the DVE and the AVE are the cells that I've been talking about all this time. They're the ones that specify where your head forms. They can be described as moving in a solid flock. So there is, as it turns out, quite a lot of mature mathematical investigations into the movement of agents behaving together as a group. Flocking behavior is the classic example where individual birds within a flock can move. And in fact, that type of maths can describe the movement of our AVE cells, where they move as what is called a solid flock. And so when I talk about solid or liquid flocks, think in terms of, say, starlings versus geese. In geese, you might describe it as a solid flock in the sense that the relative movement position of the individual geese is fixed. They do occasionally change positions, but otherwise it's fixed. Whereas starlings, it's very much more fluid, the individual birds within the flock. In our case, we think it's the math that describes it is best thought of in terms of a solid flock. The cells that move don't themselves change neighbors, but they move through a field of cells that's more liquid in its behavior. There appears to be a phase transition between the solid state and the liquid state, potentiating their movement. So I'll stop there. And I already mentioned the main people behind the work, but the main funders of this work is the Wellcome Trust for this. So thank you.
I think we've got time for one question, if people want to ask a question. Yes.
For attractor models of flock behaviour, it's helpful to have interactions between the members, for example, a constant distance between them. Do you think something like actin is true of your set of cells that behave as a flock, that there's something that binds them together independently of the other cells, and that could explain the flock-like behaviour?
It's a brilliant question. So we don't, I don't have a complete answer, but we have things which are very, very suggestive. There are various molecules that bind these cells very tightly together. So there are interactors. The notion of the elevated actin itself already starts to mechanically bind these cells together as a coherent group. So now the things that we need to, then there are other molecules which bind the cells at the basement, at the bottom, which I have an area which I didn't speak about at all. So amongst the things that we now are looking into are how these molecules are distributed elsewhere, where, as you said, one would predict there to be an attenuation of some of these molecules elsewhere in the sheet.
Thank you very much.
Thank you so much.